Good afternoon, dear students. We have been discussing principles of inheritance. In that already we have discussed about monohybrid cross. So as well, test cross, back cross, and the law of dominance and law of segregation. So today we are going to discuss about deviations of Mendelian concept of law of dominance. So already we know what is dominance. So according to Mendel, every gene has two alleles, in which one allele dominates over the other. The dominated allele is expressed as phenotype. So that is called dominance. Another allele is recessive. That did not appear in F1 generation. Only in the F1 generation we can see the phenotype of dominant allele. So that is law of dominance. Actually, we discussed about law of dominance. So Mendel factors occurs in pairs, generally which are called alleles. Factors occurs in pairs, right? So in which one allele dominates over the other. In a dissimilar pair of factors, one dominates over the other, right? So that is law of dominance. So dominance means one allele dominates over the other. Each character has two alleles. So capital T, otherwise capital T, otherwise small t. So these are all a pair of alleles which represent the same character, but which are contrasting traits. So in which capital T both are, it is homozygous. It is heterozygous condition. It is also homozygous condition. Homozygous, nothing but pure. The genotype as well, phenotype both are same. That is called homozygous. Genotype and phenotype are different. That is called heterozygous. In heterozygous condition, we can see the only tall character. The phenotype, we can see the phenotype, that is tall. But what about the small t? So that represents the dwarf character that did not appear in F1 generation. That is because of the law of dominance. But there is a deviations of Mendelian concept of law of dominance. So let's we see. In which the first one, incomplete dominance. Incomplete to dominance. When experiment conducted on pea plant, repeated with other traits and on other plants. So we had a F1 phenotype that did not resemble either of the parent. The F1 is deviated. Law of dominance. The F1 character did not resemble either of the parent is called incomplete dominance. In between them, and it is blended. The character is blended. So for example of incomplete dominance. So dog flower. Snapdragon. Snapdragon, dog flower show the incomplete dominance. So which has red flowered. red flower plant as well white flower plant. So cross it between pure red flower plant as well pure white flower plant.
Sir, sir.
So these are all homozygous. These are the parents. Parents should be homozygous. Mendel selected the parents as homozygous, nothing but pure. Mendel always selected the pure plants. Pure means homozygous. Homozygous, nothing but pure. So if they are heterozygous, continued self-pollination result in the homozygous. Continuous self-pollination result in the formation of homozygous, then selected as parents. So these are the two parents. For example, this is the female parent. This is the male parent. Right. Red flower plant is crossed with white flower plant. So gametes are formed, capital R as well, small r. These are the gametes. So F1, by crossing we get the first filial generation, capital R, small r. It is F1 generation. F1 generation. So F1 generation is always hybrid. F1 is always hybrid according to Mendel. F1 is always hybrid. But according to law of dominance, this should be red. According to law of dominance, this should be red color. But here that is deviated to pink color. So it is the pink color flowers. The plant with pink color flowers. So F1 had the phenotype that is not resemble to either of the parent, either of the male parent or either of the female parent. So pink is not present in the parents. So one parent has red flowers, another parent has white flowers. After crossing, we get the F1 generation. F1 generation show the pink color. So according to law of dominance, that should be red. But here the F1 phenotype is deviated to pink color. This is called incomplete dominance. This is called incomplete dominance. Right. F1 is self-crossed. F1 is self-crossed. Then we get capital R, small r gametes as well, capital R, small r gametes. So capital R, capital R, capital R, small r, capital R, small r, small r, small r. So this is the red color flowers. This is the pink color flowers. This is also pink color flowers. It is white color flowers. White color flowered plants. The ratio, the genotypic ratio as well phenotypic ratio. Phenotypic ratio is 1:2:1. One is red flower plant. Two is pink color plant. And one is white flower plant. Right, 1:2:1 phenotype. Phenotype means physical appearance, whatever we see in the plant. That is called phenotype. That is regulated by the genotype generally. That is regulated by the genotype. Genotype nothing but genetic makeup. Phenotype is the physical appearance. Phenotype is the physical appearance. But here the phenotype is deviated. That is called incomplete dominance. Incomplete dominance. So F1 resembles neither of the parent. F1 were in between the two parents. In between the two parents. The deviated red and white are deviated to pink. So it is called incomplete dominance. And neither allele dominates over the other. That is called incomplete dominance. So what is the genotypic ratio? Genotypic ratio also 1:2:1. 1 is Kepler, 2 is Kepler smaller, 1 is smaller. So 1:2:1 genotypic ratio. Here the important thing is genotypic as well phenotypic ratios both are same. Genotypic ratio as well phenotypic ratio both are same in incomplete dominance. Incomplete dominance show both genotypic and phenotypic ratios are same. That is 1:2:1. But in monohybrid cross, the phenotypic ratio is 3:1, but the genotypic ratio is 1:2:1. But here that is deviated. The phenotypic ratio is deviated. Genotypic is common, but the phenotypic ratio is deviated from 3:1 to 1:2:1. That is because of incomplete dominance. The example is dog flower called snapdragon antirrhinum species. Antirrhinum species, right? Antirrhinum species, dog flower, snapdragon, also called snapdragon, that is antirrhinum species. It is the example of incomplete dominance. Incomplete dominance means, so according to Mendel, law of dominance is there, but the same experiments conducted in other plants with other traits, then we see the F1, the phenotype of F1 is deviated to in between the two parental characters. In between the two parental characters. So that is called incomplete dominance. So F1 shows neither of the parent character, neither of the parental character. That is called incomplete dominance. It is one of the deviation of Mendelian. One of the deviation of Mendelian. According to Mendel, law of dominance is there. That is common. But by conducting experiments, repeated experiments with other characters in other plants, then we find the F1 is deviated from law of dominance. So the F1 shows neither of the parental character. So that is called incomplete dominance. The example is dog flower, snapdragon, antirrhinum species, right? So here the phenotypic ratio is 1:2:1, as well the genotypic ratio also 1:2:1. So both ratios are similar. It is also deviation. According to law of dominance in monohybrid cross, the phenotypic ratio is 3:1. The genotypic ratio is 1:1. Already we know. 3:1 is the phenotypic ratio and 1:2:1 is the genotypic ratio. The phenotypic ratio is deviated from 3:1 to 1:2:1. So right, it is one of the deviation. The second deviation is codominance. So right. According to law of dominance, the F1 resembles either of the parent. According to incomplete dominance, the F1 resembles neither of the parent. According to law of dominance, F1 resembles either of the parent, especially dominant parent. According to incomplete dominance, the F1 resembles neither of the parent that were in between the parents. But here, the F1 resembles both the parents. The F1 resembles both the parent. F1 both the parental characters. F1 resembles both the parental characters. So it is also one of the deviation. According to Mendel, law of dominance is there, but here F1 is deviated. The phenotype of F1 generation is deviated of law of dominance. So here F1 resembles both the parental characters. So for example, lens culinaris. Lentils. So it is the grain pulses. Widely grown in North America. So which character is deviated? The character seed coat pattern. Seed coat pattern is deviated. Which character is deviated? The seed coat pattern is deviated. So codominance means the F1 generation resembles both the parental character is called codominance. Right? In which character? Seed coat pattern. Right. So here spotted seeds. So for example, spotted seeds is crossed with dotted seeds. Spotted seeds is crossed with the dotted seeds. So these are the spots. Spotted seed. It is the dotted seed. The plant with spotted seeds and the plant with the dotted seeds. CS and CD. CS means spotted seeds. CD means dotted seeds. Right. Then we cross. So these are the parents. Parents, for example, it is the female parent. It is the male parent. So the gametes are CS and CD. So crossed, we get the CSCD. So it is the F1 generation. Right? So these are the gametes. So gametes of CS and the CD, when we cross it with these gametes, automatically we get the F1 generation. The F1 generation shows both the parental characters. What are the parental characters? And the spotted seeds with the dotted seeds. Spotted seeds, dotted seeds. Right. So it is spotted and dotted seeds. The seeds with the spots and dots, both are present in F1 generation. It is one of the deviation of Mendel. According to law of dominance, the F1 generation only shows the one of the parental character, but here the F1 generation shows the both the parental characters. Both the parental characters. Right. If we self-crossed, right? Self-crossed, the F1 is crossed with F1. CSCD is crossed with CSCD. CSCD, right. Then the gametes are CS and CD. The gametes are CS and CD. When we get to CS, 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 CD, CS, CD, CD, CD. Right, we get the 4 plants in which the phenotypic ratio is phenotypic ratio one is to 2:1 as well. Genotypic ratio also 1 is to 2:1. 1 is to 2:1. So phenotypic ratio and genotypic ratio both are same. 1 is to 2:1. One is the spotted seeded plant, another one is another two. These two are spotted or dotted seeds plants, and one is only dotted seed plant. The ratio is 1 is to 2:1. That is the phenotypic ratio. As well, the genotypic ratio is 1 is to 2:1. Genotype is CSCS is the one plant, CSCD are two plants, CDCD are two plants. So both the phenotypic ratio as well the genotypic ratio are same. So it is the codominance. Codominance. Nothing but the F1 generation shows the both the parental characters. What are the parents? And spotted seeds as well, dotted seed plants. Parents are pure spotted seeds, the plant with pure spotted seeds, as well the plant with pure dotted seeds. When we cross in between them, we get the F1 generation. We get the F1 generation. So this is the F2 generation, right? F2 generation, these are the gametes. Gametes. F2 generation, the phenotypic ratio of F2 generation is also deviated from 3:1. According to law of dominance, the F2 phenotypic ratio should be 3:1. But here that is deviated into 1:2:1. So it is the deviation of Mendelian, that is co-dominance. Co means the similar, and F1 generation show both the parental characters. So that is called co-dominance. These are the two deviations of Mendel. These are the two deviations of Mendel. One is incomplete dominance. In incomplete dominance, the F1 is deviated. F1 shows neither of the parental characters. F1 were in between the two parents. Between the two parents. So right, incomplete dominance. So here the example is dark flower snapdragon, that is called anti-rhinum species, right? So here red flowered plant is pure red flowered plant is crossed with white flowered plant. Automatically we get the F1 generation. That is pink color plant. Pink is not there in the parents. Pink is not there in either parents. One is red, another one is white. But the F1 is pink. So that is the deviation of Mendel law of dominance. That is self-crossed. Then we get the F2 generation. Right. In F2 generation, we get the ratios, phenotypic ratio, that is 1:1, as well the genotypic ratio, that is 1:1. Both are same. According to law of dominance, phenotypic ratio should be 3:1. But here, that is deviated to 1:1. So incomplete dominance and codominance in both conditions, the phenotypic ratio is 1:2:1, as well the genotypic ratio is 1:1. These both are common. It is very, very important. It is very, very important. Right. These are the two deviations of Mendel. Next one, pleiotropy. Pleiotropy. Single gene product may produce more than one phenotype. More than one effect. Single gene product may produce more than one effect. Single gene produce more than one phenotype. One phenotype. Generally a gene nothing but, so this is the chromosome, it is the DNA. A segment of DNA is gene, that is the gene. Generally the gene converts into mRNA. mRNA converts into a protein. Protein may become multiple enzyme, hormone, receptors, antibodies, antigens. So everything is the product of a protein. So this we can see. These are the phenotypes. These are all phenotypes. So this is the genotype. This is the genotype. But we are diploid. we have two chromosomes for each character. We have two chromosomes for each character. This is one chromosome. For example, number one chromosome, it is also number one chromosome. It we get from father, it we get from mother. So we got the two chromosomes, one from one parent, other from other parent. So this is also same gene, but different forms. Those are called alleles. Each gene has two forms. Those are called alleles. These are the two alleles which are present on the locus. It is called locus. On the locus, two alleles are there. Two alleles, nothing but alternative forms of a gene. Alternative forms of a gene are called alleles. So this is totally a gene that has two copies. One copy present on the father, another copy present on the mother chromosomes. These are the two chromosomes we get from parents. Our father gives one set of chromosome, that is called one genome. Our mother gives us one set of chromosome, that is also one genome. And a haploid set of chromosome is called genomes. So then we get to 23 chromosomes from father, as well 23 chromosomes from mother. We get 46 chromosomes, 23 from father, 23 from mother. So the first chromosome of father and mother constitute the homologous chromosomes. They have the same genes, they express the same characters. They have same genes and they express the same character. So this is called gene. Right? Gene has alternative forms. Those are called alleles. Alleles are alternative forms of a gene which are present on homologous chromosomes. Homologous chromosomes. Now, what is the duty of gene? Gene expresses in the form of phenotype that we can see. Only we can see the phenotype. We can't see the genotype that is present in the chromosome. Genes we can't see, but only we can see the gene product. That is the phenotype. Phenotype, right? Protein converts into different phenotypic forms. That may be enzyme, that may be hormone, that may be enzyme, that may be hormone, that may be antigen, that may be right receptor, that may be pigments. So everything is the product of a gene. Only we can see the color, we can see the height, we can see the hair. These are all phenotypic characters that we can see. Right here, only one gene can express one character generally. Single gene affect only one character. But in this condition, in pleiotropic condition, single gene product may produce more than one effect. That is nothing but more than one phenotype. Single gene produce more than one phenotype. That is called pleiotropy. So, for example, starch grain synthesis of pea seeds. Starch grain synthesis of pea seeds. Right. That is controlled by a gene that has two alleles. Those are capital B and small b. The alternative forms of a gene is capital B as well, small b. These are the two alleles of a starch, the gene of starch gain synthesis, which regulate the synthesis of starch. These are the two alternative forms of a gene of starch grain synthesis, right? Capital B and small b. So we take the capital B, the seeds with capital B is crossed with the small b. Automatically we can get the capital B, small b. So right, it is the F1 ratio. Automatically when we crossed self-pollination, self-crossing, automatically we get the capital B, small b, or the gametes as well, capital B and small b. Then we get the capital B, capital B, capital B, small b, capital B, small b, small b, small b. So here the ratio is F1 is always 100% hybrid. Right here, capital B, capital B. It shows the large starch grains. Large starch grains. Capital B, small b is 1. These are the two intermediate starch grains. And small b, it is one plant. It shows small starch grains. Small starch grains. Right. It is one of the characters. Starch grain size is one of the character. Here, what are the two characters? Single gene product may produce more than one effect. That means single gene controls two characters. Single gene controls two characters, two phenotypic characters. What are the two phenotypic characters? One is starch grain size. Size of starch grain. The second character is shape of the seed. So right here capital B, but the seed is round. But the seed is capital B contains large starch grains with a round shape, with a round seed. Capital B, small b. Intermediate starch grains, but the shape is round, round seed. And small b, small starch grains as well. The seed shape is wrinkled seed. The ratio is, the phenotypic ratio is 3:1. Round seeds are 3 and wrinkled seeds are 1. Round seeds are 3, wrinkled seeds are 1. Here starch grain size, one seed is large starch grains as well. Two seeds are intermediate starch grains. One seed is small starch grains. This is one character. One character. It is the second character. These two characters can be controlled by the single gene. Single gene controls the two characters. So one is the starch grain size. Another one is the seed shape. Another one is shape of the seed. These are the two characters controlled by the single gene. Generally, according to Mendel, single gene controls one character, but here single gene regulates the two characters. So one character is the starch grain size. Starch grain size. Starch grain size. The second character is the seed shape. So here the first character, that is the starch grain size, shows the incomplete dominance. Incomplete dominance, nothing but in F1 generation, capital B, small b shows the intermediate starch grains. Intermediate starch grains. It is not present in the parents. Parents have capital B, small b, small b. It is the large starch grains, it is the small starch grains. No intermediate starch grains present in the parents. But in the F1 generation, shows the intermediate starch grains. It is not present in the parents. It is deviated. So that is the character shows the incomplete dominance. Incomplete dominance. Incomplete dominance. The character, that is the starch grain size, is the incomplete dominance. That shows the incomplete dominance. As well, the second character, seed shape, shows the law of dominance. It shows the law of dominance. Follow the law of dominance. So the hybrid condition shows the one of the parental character, that is round seed. Round seed is present in the parent, that is appeared in the F1. So F1 is similar to one of the parent. So right, that is the dominance character, that is law of dominance. It is incomplete dominance. These two characters are regulated by the single gene. So that is called, that condition is called pleiotropy. What is pleiotropy? Single gene may affect the more than one phenotypes. Single gene affect the more than one phenotype, right? Single gene product may produce more than one effect, nothing but one, more than one phenotype. It is the pleiotropy. Very, very important concept, pleiotropy. Next, concept of law of dominance. Concept of law of dominance. Concept of law of dominance. So what is dominance? Why some alleles show dominance over other? Why some alleles show the dominance over other? To tackle these questions, we must understand the what a gene does. What a gene does. Right, it is very important question. What is the duty of a gene? So what is dominance? We see the dominance. One allele dominates over the other. Why some alleles show dominance? Why some alleles are recessive? Why some alleles are recessive? Why some alleles are dominant? Why some alleles are recessive? Right, to tackle these questions, we must understand the gene. What a gene does. So gene nothing but express the phenotype. A gene is always contains the information. A gene always contain the contains the information to express a trait. To express a trait. A gene always contains the information to express a trait. That is the gene. Gene contains the information to express a phenotype or to express a trait. Right. Each gene has two alleles. Each gene has two alleles. Already we know. For example, the gene for height. The gene for height has two alleles. Those are capital T, otherwise capital T, small t, otherwise small t, small t. Capital T, capital T, capital T, small t, small t, small t. So these two are the alleles of a same gene. These two also alleles of a same gene. These two also alleles of a same gene. Nothing but the gene that regulate the character of height. Height is regulated by the gene that has two alleles, two alternative forms of a gene. Right, these are the two alleles in which one allele dominates over the other. Why one allele dominates over the other? Right. One allele dominates over the other. So, right, example, based on the example, enzyme producing gene. We take the an example of enzyme producing gene. So a gene can produce an enzyme. And the enzyme is the phenotype, the gene is the genotype. A gene produces enzyme as a phenotype. Enzyme producing genotype, right? Here two alleles are there. So for example, two alleles are there. For example, we take the one allele and another allele, that may be small or that may be capital. And this enzyme producing gene has two alleles. Enzyme producing gene has two alleles which are alternative forms. Two alleles which are alternative forms in which one allele is unmodified allele. Unmodified allele that expressed to produce an enzyme. That expressed to produce an enzyme. Produce an enzyme. It is responsible for transformation of substrate into product. So what is the duty of enzyme? An enzyme converts the substrate into product. An enzyme converts the substrate into product. Right, this is the enzyme. Enzyme converts the substrate into product. Right, substrate into product. What is the enzyme duty? So enzyme is produced by a gene. Enzyme is produced by a gene. It is the phenotype, it is the genotype. Genotype and phenotype. That is the enzyme is the phenotype that transform the substrate into product. Right. It is unmodified allele. What is unmodified allele? Right. Unmodified allele can produce an enzyme that transforms the substrate into product. And the other allele may be modified allele. Other allele may be modified allele. The modification could be could be produced right. One normal enzyme less efficient enzyme or non-functional enzyme no enzyme. So these are the three forms produced by modified allele. Modified allele. Right, unmodified allele always expressed. Unmodified allele always expressed. But expressed to produce the enzyme. Right. That is always expressed. But the other allele may be modified. The modification could be normal enzyme or less efficient enzyme or non-functional enzyme or no enzyme, in which there are three conditions. In the first condition, normal enzyme is produced by modified allele. So automatically, unmodified allele is the T. It is responsible for the expression of phenotype. Unmodified allele always expressed and produce the phenotype. Right? It is the unmodified allele. As well, the another allele also produce normal enzyme. That is nothing but equal to unmodified allele. So these conditions we can see more. These conditions are common, right? Then it becomes homozygous. Here both alleles are responsible for the expression of a phenotype. Both alleles are the expression of a phenotype, are involved in the expression of a phenotype, right? It is unmodified allele. It is modified allele. Modified allele is equal to unmodified allele. So both can produce the enzyme, right? And second condition, non-functional enzyme. Here unmodified allele is there, but the second allele is produce non-functional enzyme like this. Capital T, small t, for example, right? Here heterozygous condition is there. Heterozygous condition, it is the modified allele that is not able to generate enzyme. Then in this condition, only unmodified allele is responsible for the production of enzyme. Right here, this is the dominant, this is the recessive. Recessive nothing but the allele does not express. That is recessive, right? In third condition, no enzyme like this. Here also, this is unmodified allele. This is the modified allele. Right? In which condition also, unmodified allele is the responsible for the production of a enzyme. Modified allele does not express. It is the first condition. It is the second condition. It is the third condition. What about small t, small t? Small t, in which condition both are modified alleles. They both don't express the phenotypic character. So, right, then homozygous recessive condition appears. Homozygous recessive condition appears whenever the two modified alleles are present. Recessive, nothing but they don't express. Both the alleles don't express. That condition is called recessive. So it is the concept of law of dominance. Law of dominance means already we know what is dominant, what is recessive. So what is dominance? In hybrid condition, one allele dominates over the other. The dominated allele is called dominant allele. Other allele is called recessive allele, right? Why some alleles show dominance over the other? What a gene does? Why some alleles are recessive? Why some alleles are recessive? Why some alleles are dominant? Right. Right. Then we have a question, that is what a gene does. What a gene does? A gene always contains the information to express it right. To express it right. Each gene has two alleles. Each gene has two alleles. Because we are diploid. Because we are diploid, each gene has two alleles. In which one is modified allele, another one unmodified allele. Modified allele is always responsible for the production of a phenotype, but modified allele sometimes may express, sometimes may not express, because right here two alleles are there. One is unmodified allele, that is always expressed to produce an enzyme. The other one is modified allele. Modified allele may be in three forms, in which one condition is not that produce the normal enzyme, that is equal to unmodified allele. The second condition is non-functional enzyme, and that is also modified allele. And the third one is no enzyme. And modified allele can produce normal enzyme sometimes, can produce non-functional enzyme, can produce no enzyme. In these conditions, only unmodified allele is responsible for the production of a phenotype, that is nothing but enzyme. Right? Here both are equal. That is nothing but the other allele is also produce a normal enzyme. So in which condition they are homozygous? This condition is common, right? And the second condition, one is unmodified allele, the second one is the modified allele. That condition becomes heterozygous, in which unmodified allele is responsible for the production of a phenotype, nothing but here enzyme. And the third condition also, one is non-functional enzyme, no enzyme, no enzyme. In that condition, the unmodified allele is responsible for the production of a phenotype, nothing but here enzyme. So this is the concept of law of dominance. Concept of law of dominance. Okay, we have completed today the deviations of Mendelian. Deviations of Mendelian concept of law of dominance. So in which the first one is incomplete dominance. Incomplete dominance means the F1 progeny does not show any parental character that were in between them. Here the red flower plant is crossed with white flower plant, automatically we get the F1 generation. F1 generation show the pink one, pink colored flowers. The F1 is self-feed. Then we get 4 plants. The phenotypic ratio is 1:2:1. The genotypic ratio is also 1:2:1. So one is the red color plant, 2 are pink color plants, 1 is white colored plants. So here hybrid condition is deviated. Law of dominance. Hybrid conditions show neither of the parental characters that were in between the two parents. and the codominance. Here the F1 shows both the parental characters. Spotted seeded plant is crossed with the dotted seeded plants. Automatically, F1 generation shows spotted and dotted seeded plants. Here the F1 shows both the parental characters. So it is also deviation, right? Then next we pleiotropy. Single gene product may produce more than one effect, nothing but more than one phenotype. So one gene controls the more than one phenotype. Right here the two phenotypes are, one is the starch grain size. Starch grain size is one of the character. Another one character is the seed shape. So these are controlled by the B gene, capital B and small g. These are the two alleles. When we cross in between these two, automatically we get the 4 plants in F2 generation. Capital B shows the large starch grains, but the seed shape is round. Round seed, large starch grains, capital B, small b, capital B, small b. These two plants show the intermediate starch grains with round seeds. Small b shows the small starch grains with wrinkled seed. So here the seed shape follow the law of dominance. So here 3:1 is the ratio is there. 3 plants having the seeds with round, 1 plant having the seed with wrinkled. In the starch grain size, 1 seed having large starch grains, 2 seeds having intermediate starch grains, 1 seed having small starch grains. Here the intermediate starch grains are appeared. That is the incomplete dominant character. Starch grain size is the incomplete dominance, and the seed shape is the law of dominance character. So this is the very, very important topics. Dear students, if you got any doubts, call to the number scrolling in the screen.
ఎవరికి ఏం డౌట్లు ఉన్నా దయచేసి జూమ్ లో జాయిన్ అయిన వాళ్ళు అన్మ్యూట్ చేసి మీ డౌట్స్ ని సార్ని అడిగితే సార్ మీ డౌట్స్ క్లారిఫై చేస్తారు. దయచేసి జూమ్ లో ఎవరైతే జాయిన్ అయ్యారో మేము లింక్ ఇచ్చాము, జూమ్ ఇచ్చాము. దయచేసి మీ డౌట్స్ క్లారిఫై చేయడానికి జూమ్ ఇచ్చాము. మీ డౌట్స్ ఏ ఉన్నా కానీ మీరు అన్మ్యూట్ చేసి దయచేసి సార్ని అడిగినట్లయితే సార్ మీ డౌట్స్ అన్ని క్లారిఫై చేస్తారు. Once again, I repeat, whoever have doubts regarding the topics which have been covered as of now, please unmute yourself. We have given an option that you can unmute yourself. So please ask the questions. మీరు అన్మ్యూట్ చేసుకున్నట్టయితే మీకు అన్మ్యూట్ ఆప్షన్ ఉంది. యా భద్రాచలం.
Dear students, one small topic is there. Here, so here law of dominance is deviated. So dominance is not a autonomous feature. According to law of dominance, dominance is autonomous feature, but here dominance is not an autonomous feature. That depends on the gene product as well the phenotype. Gene product and the phenotype. Dominance is depends on the gene product and phenotype. That is not an autonomous feature, right? Because here incomplete dominance and codominance is there. We have seen the incomplete dominance as well. We have seen the codominance as well. Seen the starch grain size is intermediate condition, which are all the deviations of dominance. Based on that, dominance is not a autonomous feature. It only depends on the product of a gene as well the phenotype. Product of a gene as well the phenotype, right?